Well, good afternoon, everybody. How are you doing today? Is the sound clear on your side? Can you hear me? Yes. Great. All right, so we are discussing, as you remember, the electrical conduction through the nerves. If you remember, any electrical signal is going to be in the form of change in the state of polarity of the membrane of the cell. If you remember, if I'm looking at any cell, you're going to have different ion distribution across the cell membrane. So if this is a nerve fiber, for example, or a muscle cell. So what we're looking at in here, this is going to be the cell membrane. This is my intracellular fluid, the fluid inside the cell, which is the cytoplasm. And we call the fluid outside of the cell, this is my extracellular fluid. If you remember, inside your cells, you're going to have much more anions, much more negatively charged ions compared to the outside. This doesn't mean that you only have negatively charged ions on the inside of the cells. This simply means that you have more negatively charged ions inside than outside. What is the major cation located inside your cells in the intracellular fluid compartment? Inside the cells, I have potassium. Potassium is going to be present inside the cells at much greater concentration levels compared to the outside of the cell. Compared to outside of the cell, the major cation of the extracellular fluid is going to be my sodium. So again, the ion distribution across the cell membrane, inside of the cell, I have much more negatively charged ions. Those are called my anions compared to the outside. The major cation, the major positively charged ion located inside of the cell, this is going to be my potassium. And the major cation outside of the cells in the extracellular fluid, this is going to be my sodium. So knowing this, as I have much more negatively charged ions inside the cell, so the inside of the cell membrane is going to be more negatively charged than the outside of the cell membrane, which is going to be more positively charged. I didn't say that this is purely positive, this is purely negative. I'm saying that if you compare them to one another, the inside of the cell membrane is going to be more negatively charged compared to the outside. So we decided to measure, if you remember last time, we measured the difference between the inside and the outside to check the difference between inside and outside. We'll find out that inside is more negatively charged than the outside by a value of 70 millivolts. So inside is more negatively charged, and the electrical polarity on here during rest. So here we're looking at in resting conditions. So during rest, inside is more negatively charged than the outside by a value of 70 millivolts. So the resting membrane polarity, what I mean by resting membrane polarity, Any idea? Any idea? What is the resting membrane polarity? As the value of the resting membrane polarity is negative 7 millivolts, but what is it by definition? It is the difference in charge between the inside of the cell membrane and the outside of the cell membrane. When? During rest. If you're not conducting any signals along the plasma membrane, this means that the resting membrane polarity is going to be equal to negative 70 millivolts. It's not neutral. It's not neutral. It's negative 70 millivolts. I'm not conducting anything. It's not neutral. Inside is more negatively charged by a value of 70 millivolts. Did you guess this, Shannon? All right, so here the cell is not conducting any electrical impulse. I'm not conducting any electrical impulse. So if I'm drawing this on a graph and I'm plotting on the graph on here, the voltage and time. This is what I'm drawing on the graph. The relationship between the voltage and time. So here, if I'm not conducting any electrical impulse, I don't have any stimulation of the nerve cell. When I check the membrane polarity, it's going to be negative 70 millivolts. This is in resting state. I didn't receive any kind of stimulation to the nerve. So the nerve is not going to be conducting any electrical impulse. What if you have received a stimulus? Like what? Like, for example, you applied enough pressure on your skin, so your touch receptors will start to send out a signal to your brain for you to feel that you're touching something. For example, if you are hearing my voice right now, this means that my voice is stimulating the hearing receptors in your ears, and they will start to generate an electrical impulse that will be traveling to reach your brain for your brain to decode those signals into something that you can understand. So again, my resting membrane polarity is equal to negative 70 millivolts. When would I be sending out an electrical impulse that will be traveling for long distances? We call the electrical impulse that gets formed if you've got enough stimulation. And remember, what do we call the minimal stimulus that will be forming this electrical impulse that will travel for long distances? We call the minimal stimulation here, it's my threshold stimulus, exactly. So if I had as a threshold stimulus, can I have a stimulation without conducting the signal to my brain? What do you think? Is there any time when you're going to be receiving a signal, but this stimulation, you are exposed to a stimulus, but this stimulus will not be translated into an electrical impulse that will reach your brain? What do you think? Is this possible? What do you think? Can I get exposed to a stimulus which is lower than the thresholds, and this won't form any electrical signal for my brain to decode? No. So for example, this is my eye here. As we're going to learn later next week, the light is going to be traveling like this. And those light rays going to be converged for them to be focused on the retina in the eye. This is where I have the receptors responsible to create an electrical signal to be sent to the brain for you to be able to see. Can I have light rays coming in my eyes, but those light rays are not going to be detected by those receptors? What do you think? Can you see every single color normally if you don't have any problem with your eyes? Can you see every single light? What do you think? No, you can't, right? So for example, you have a remote control for your TV. Is there any light that gets emitted from this remote control? Yes. Can you see the light coming out? Not is it on the TV itself. Can you see the light ray emitted from your remote controller? No, you can't. Why? Because this light is infrared. This is below the spectrum that will stimulate those receptors to create an electrical signal to be sent to my brain. Do they enter my eye? Do I have infrared light coming into my eye? All the time you get infrared. Can I see them? No, you can't. Why? Because they are gonna be performing a stimulation below threshold. Clear, clear? So in order for me to create an electrical signal that can propagate along my nerve cell to reach my brain from a receptor, for example, you're gonna need to get a minimal, at least a minimal stimulation, which is my threshold stimulus. I receive the threshold stimulus. How would the nerve cell respond? Nerve cell creates an electrical signal by changing its membrane polarity. So this is my cell on here. This is the same cell. After it gets exposed to a stimulus, how would I respond? I'm trying to change the membrane polarity. What I mean by flipping the membrane polarity, this simply means that rather than inside to be negatively charged, more negatively charged, outside will be more negatively charged. And the inside is going to be more positively charged. This is my electrical impulse. This is the electrical signal that you can send right now. Yes, so the appropriate receptor, what will make a receptor appropriate for a signal and not for another is its threshold. So if the stimulus, if for example I did have, let's say, a receptor in my eyes that can detect infrared, so this receptor is going to be adapted to create, for it to have enough sodium into the cell at a lower threshold. So this means what? This means that my receptor, because why is it not adapted to detect infrared stimulation? Simply because it's not a threshold stimulus for it. It's lower than the minimal stimulus that it requires in order to conduct an electrical signal. Does this make sense, Hershey? So for example, why would some animals detect sound waves that we can't hear? Because they have modified cochlea and receptors detecting wavelengths that we do not detect. They have the ability to detect other frequencies that we don't have the ability to detect, which will make those frequencies threshold for them, but not for us. All right. So again, simply speaking, how would I be sending out the electrical signal along my nerve cell? I will lose the membrane polarity. What was the membrane polarity? Inside was negatively charged, outside was more positively charged. So in order for me to lose the membrane polarity, I will flip the charge. Outside should be more negatively charged, and inside should be more positively charged. So what we're concerned with, what we are always measuring here, is inside. So in a resting membrane polarity, what was the voltage of the inside of the cell membrane? It was negative 70 millivolts. So now, if I'm drawing this, I did receive a stimulus, and this stimulus did make the inside of the cell membrane to become more positively charged. So this means that am I going up or down on the graph? This is my 0 on here. Would I be going up or down on this graph? I'm going up. So there will be an increase in the membrane voltage. So rather than being negatively charged on the inside, it's going to become more positively charged on the inside. So I start with negative 70, I go all the way up. Once I reach enough voltage, I need to go back to my resting membrane polarity. I need to reach back to become negative 70 millivolts. So for you looking on here, what was the ion, if you remember from last class, what was the ion responsible to allow the cell to create this electrical signal? Sodium. So if I get enough stimulation, I will keep opening sodium channels, allowing the sodium down the concentration gradient. Remember, sodium has a very high concentration outside of the cell compared to inside of the cell. So once you open the sodium channels, what's going to happen? The sodium will pass to the inside of the cell, and this will make the inside of the cell membrane to become more positively charged. If I get another stimulus at this time, would I be able to conduct another electrical impulse? Remember, can I open a door that is already opened? No, I can't open a door which is already open. If I am already positively charged, I can't become positively charged while I am already positively charged. The electrical voltage here, it's all or none, whether you get it or not. So you can't increase the amplitude. This won't happen in the formation of an action potential. So if you get another stimulus here, you will simply neglect it. You won't respond to this stimulation. Why? Because this stimulus did take place while the membrane is already more positively charged. We call the state in which I am losing my membrane polarity. Inside is becoming more positively charged than the outside. This is my depolarization. Remember, D, it's the opposite. I've lost my polarity. So we call this phase as my depolarization phase. All right. This is my depolarization phase. In order for me to respond back, to be able to start to respond to other stimuli, if I received any, I got another stimulus on here. In order for me to respond to this stimulus, I need to go back all the way down to my resting membrane polarity for me to start again another electrical impulse. You need to close the door in order to reopen it again. You can't open an open door. So here in a depolarized phase, in a depolarized membrane, you can't perform any further depolarization. It's all or none, whether you are depolarized or you're not. I can't depolarize an already depolarized cell membrane. So in order for me to repolarize, to return back to the polarized state, what should I be doing? I need first to stop the influx of the sodium. So it doesn't make any sense that you keep the sodium moving in while you're trying to become more negatively charged on the inside, returning back to your negative 70 millivolts. So this outer gate is open to allow the sodium in. In order for me to stop the influx of the sodium, what should I do? I need to close the internal gate for the channel. So I close the sodium channel. And this is going to be the first thing you need to do. And this is not going to be enough. You prevented any further influx of the sodium, doesn't make you more negatively on the inside. You need also to get rid of some of the positively charged ions on the inside of the cell membrane. You need the inside of the cell membrane to lose positively charged ions. So if I did try to move the sodium, remember, outside of the cell, you've got much more sodium than inside of the cell. You did allow a very small amount, of sodium in the cell to allow the depolarization to take place. So this is not gonna be helping you moving the sodium out to become more negatively charged another time. Why? Because you are trying to move the sodium against the concentration gradient. So why don't you move the sodium by active process? So you spend energy to move the ion against its concentration gradient. it's going to be time consuming. It's going to be much slower. So I need to find another ion that is already present on the inside and is positively charged. And for me to allow, once I allow it out, I return back to my resting membrane potential. So what is the ion that you already have a lot of it on the inside of the cell? Is your potassium. So in order for me to become negatively charged, again, I will open the potassium channels. And once you open those potassium channels, the potassium will be moving from its high concentration gradient inside the cell to its lower concentration gradient outside of the cell. So potassium here is going to be moving down the concentration gradient. This is potassium efflux. I am moving out of the cell. So once you lose enough positively charged ions from inside of the cell, what's going to happen to the inside of the cell membrane? Will it become more negative or more positive? What do you think? If I'm losing positively charged ions from inside of the cell, will the cell membrane become more negative or more positively charged? More negative, exactly. See on here, I stopped the influx of the sodium and I started to allow potassium out of the cell. The potassium efflux, you're allowing the potassium out, allows you to return back to the polarized state that you started from, to the negative 70 millivolts. So what do we call this phase of the electrical signal, of the action potential? When you return back to the polarized state, this is my repolarization. This is what we discussed last class. We stopped at this point on Tuesday, if you remember. Any problem? Any problem so far? So, Christina, I don't understand, what did you mean by vesicles? Yeah, so this is what we're going to be discussing in a minute. This is what we're going to be discussing in a minute. All right, so Christina here is saying, so if I'm allowing sodium in, so over time, the sodium is going to build up in the cell and there will be no concentration difference. So the sodium concentration gradient will be disturbed. So you won't have any more sodium outside as inside. All right, so because over time, every time you're going to be conducting an electrical impulse, some of the sodium going to be moving in, so the sodium builds up in the cell. So this is a question. This is a really good question here. All right, so we're going to answer this in just 1 minute. All right, so I did allow the sodium in. I did allow the potassium out, and again, when you allow the potassium out of the cell, the inside of the cell will return back to its polarized resting membrane polarity. So inside is going to become more negatively charged another time, and the outside will become more positively charged than the inside another time. Again, what do we call this phase? This is my repolarization phase. What if I told you that the potassium channels will not immediately close? It's going to take them a while to close, even after you reach the resting membrane potential. So I did reach already the resting membrane potential. I did reach already negative 70 millivolts. This is my repolarization, but the potassium channels are gonna still be leaking potassium out of the cell, even after you get back to your resting membrane potential. I'm allowing more potassium than what I need out. So what's gonna happen to the cell membrane polarity? Inside will even become more negatively charged than the resting membrane potential, as you kept losing potassium to the outside. You see on here, you kept losing more potassium even after you reach the resting membrane potential, and the membrane will become even more negatively charged than the resting state. We call this phase of the action potential, it's my hyperpolarization. What I mean by hyperpolarization, I'm even more polarized than my resting membrane polarity. I'm even more polarized, hyperpolarization, extra polarized. I am more negatively charged. So back to the question here from Christine. So if I am allowing the sodium in and allowing the potassium out, so over time, if I'm not doing anything about it, the sodium is going to be building up inside the cells, and the cells will be losing their potassium to the outside. But let me ask you a question first before I answer this. Do you remember, in order for your brain to function normally, it needs continuous supplies of what? You remember from our last meeting? What does your brain require, or your nerve cells require, to maintain their function? I need continuous supplies of glucose. I need continuous supplies of oxygen. Remember, if I have hypoxia, I have low oxygen, I might lose consciousness. My brain won't able to function normally. If I have hypoglycemia, if I have a drop in my blood sugar level, my brain won't function normally. I'm not conducting electrical signals. Why did we justify the need of continuous supplies of oxygen and glucose to the brain? We justified this, but as the brain has a very high metabolic activity. It requires a lot of energy to conduct its action. All right, so did we consume any ATP here to create this electrical impulse? Do you see any consumption of ATP? Did we say anywhere today that we are in need of an ATP? What do you think? No, we didn't mention any ATP. The ATP is going to be needed to maintain the concentration gradients. So remember, inside of the cells, you're going to have much more of the potassium. And outside of the cell, you're going to have much more of the sodium. So in order to keep the concentration gradients this way for both the sodium and the potassium, what will I need to do? I need to move sodium outside of the cell back to the extracellular fluid. And you see on here, I am moving the sodium from its low concentration gradient to its high concentration gradient. In the same time, I need to move back the potassium that I allowed outside during the repolarization and hyperpolarization. I need to get it back also against the concentration gradient, because I still gonna have high concentration of the potassium inside of my cells. How would I be able to allow this to take place? If you remember, we did study back in the cell chapter a type of active transport which we called the sodium potassium ATPase pump. And if you remember, this pump did have binding sites. Here I am looking inside the cell. This is outside of the cell. The pump is normally facing the inside of the cell. It's going to be binding to 3 sodium. And it needs, in order to move them out of the cell, it needs to hydrolyze an ATP. So it has a site where the ATP gonna be binding and gets hydrolyzed. The hydrolysis of the ATP will be releasing energy that will change the shape of the pump. This is a protein like the egg white. So what happens to the egg white when you heat it? When you heat a raw egg, what happens to the egg white? Rather than being liquid, rather than being clear, it's going to become white and solid, right? It's going to be denaturated. Here is something similar. It's not a denaturation of the protein. It's not a permanent change in the protein, but it's a modification in the shape that results from the hydrolysis of the ATP. So when I break down the ATP into ADP and an inorganic phosphate that remains attached, this will cause a change in the shape of my pump. And this change allows the pump to move the 3 sodium out of the cell. So you see on here, I did rotate. Those are the 3 binding sites of the sodium. They are now facing the outside. And they will be releasing the sodium outside of the cell. So where the sodium did move, it moved from inside of the cell. This is the inside again, and this is the outside. So I did move the sodium from inside of the cell to the outside of the cell. Then, as I release those 3 sodium and the inorganic phosphate from the pump, this will expose 2 binding sites for potassium that will be binding to potassium like this. As you get those 2 potassium attached to their binding sites on the sodium potassium ATPase pump, the pump rotates back to its normal shape, facing inside. So again, how will it look like? Looking on here, again, this is inside, this is outside of the cell. and this is my pump. The pump would be rotating back, facing the inside of the cell, and bringing the 2 potassium from outside in that will be released to the inside of the cell. When this takes place, when would you activate the sodium potassium ATPase pump, do you think? After how many action potentials you conduct? What do you think? How many action potentials you're going to be conducting through the nerve cell, for example, to activate the sodium potassium ATPase pump? Just guess. I'm waiting for any guesses here. Unlimited. So you don't require it, or you're going to need it all the time, regardless of the action potential? After each one, you're going to start to activate. Actually, sodium potassium ATPase pump is going to be pulling the sodium out and moving the potassium in all the time. All the time, you're going to be trying to move more sodium out, trying to bring more potassium in. All the time, all the time. In the background of the action potential, in the background, you're going to be activating the sodium potassium ATPase pump. And this is actually what will be creating this difference in the concentration. So whenever you need to depolarize, you've got the sodium outside at high concentrations. Whenever you need to repolarize, you've got the potassium inside of the cells to repolarize. All right. So again, when would the sodium potassium ATPase pump gonna be activated? It's always activated. And that's why, if you remember, brain needs continuous supplies of sodium and potassium. I'm sorry, of glucose and oxygen. All the time, your brain gonna need to generate energy, and this energy is gonna be needed for the sodium potassium ATPase pump to be activated. All right, questions? All good? All right, so let me pull a video here, video animation that shows the steps of the action potential.
How does the pain you experience when you burn your hand result so quickly in an action by your muscles? Many animals respond to environmental stimuli using specialized cells called neurons. A stimulus is detected by sensory receptors, and the body responds through motor effectors. These cells working together allow you to respond very quickly to threats. When you touch something hot, heat receptors of a sensory neuron detect the stimuli and send the information of heat to an interneuron in your central nervous system. From there, a motor neuron sends a response from your central nervous system to the skeletal muscles in your arm, causing them to contract and pull your hand away. The fundamental process of neural transmission that underlies this action occurs in all neurons of the body. Neurons transmit this information through changes in the electrical potential of the membrane by the movement of ions across the membrane. An electrochemical gradient governs the movement of these ions, resulting in an electrical impulse. The resting membrane potential in a neuron, when the cell is not firing an impulse, is established by the unequal distribution of sodium ions outside of the cell and potassium ions inside the cell, making the outside of the cell more positively charged compared to the inside. The electrochemical gradient is established and maintained by an enzyme called sodium-potassium Atpase. When a neuron is stimulated, sodium ion channels open and sodium ions flow into the cell. This leads to a change in the electrical potential across the membrane called depolarization. The depolarizing electrical potential travels down the dendrites and over the cell body. Multiple electrical potentials will combine at the axon hillock in a process called summation. If the depolarization is large enough, an action potential is triggered. Action potentials are all or none electrical impulses that maintain their amplitude and strength down the length of the axon. The action potential travels down the axon when the depolarization of an area of membrane causes adjacent voltage-gated sodium ion channels to open. The influx of sodium ions results in membrane depolarization along the membrane. After a short delay, potassium ion channels open and potassium ions flow out, repolarizing the membrane. For the neuron to fire again, the resting membrane potential needs to be reestablished. Sodium-potassium ATPase is used to move sodium and potassium ions against their concentration gradients, reestablishing the resting membrane potential. As the action potential moves down the axon, ions are diffusing only a short distance, allowing the signal to move quickly. At the axon terminal, the electrical impulse passes to another cell at a cellular connection called a synapse. The space between the presynaptic neuron and a postsynaptic cell is called the synaptic cleft. The presynaptic neuron contains signal molecules called neurotransmitters that are packaged inside vesicles. When an action potential reaches the end of a neuron, neurotransmitters are released by exocytosis from the neuron into the synaptic cleft. Neurotransmitters bind to the adjacent cell at receptor sites attached to ion channels. The channels open, allowing the movement of ions into or out of the effector cell, which alters its membrane potential, thereby transmitting the signal from the neuron to the effector cell. Because nerve impulses move very rapidly down the axon of a neuron and move from cell to cell across synapses, you react quickly to a stimulus, like burning your finger.
So again, one more time before we move on. In a resting state, if I'm not perceiving any kind of stimulation, if I'm not detecting any kind of stimuli, what's gonna be my resting membrane potential? Inside is gonna be more negatively charged compared to the outside, around negative 70 millivolts, due to the presence of much more anions inside my cell. If I got enough stimulation, what's gonna happen? I will be opening enough sodium channels that will be allowing the sodium to move down their concentration gradient. Sodium, as it moves from outside of the cell to the inside of the cell, inside of the cell will become more positively charged. So I've lost my membrane polarity. We call this is my depolarization. And again, a very common type of questions that you will see is which of the following is responsible for depolarization? Sodium influx, sodium influx, potassium influx, potassium influx, calcium influx. So which one do you think? Hello? Sodium influx. Sodium influx. As I open enough sodium channels, I will reach the apex of my action potential. It can go beyond this point. I can't keep going up. So at this point, what's gonna happen? Sodium channels gonna close, and I will open potassium channels for me to return back to the polarized state, to the resting membrane polarity. When I open up those potassium channels, the potassium is gonna be traveling again down its concentration gradient from inside of the cell to the outside of the cell. And as a result, inside will become more negatively charged again, but something that we need to be aware of that those potassium channels are slow to close. So they won't close immediately when you reach negative 70 millivolts. They will be leaking some of the potassium outside of the cell, bringing the membrane polarity to become more negatively charged, even more negative than the resting membrane polarity. We call this phase of the action potential, it's my hyperpolarization. What maintains the ion concentration this way? What keeps the sodium at high concentration gradient outside of the cell and keeps the potassium at high concentration gradient inside of the cell is going to be my sodium potassium ATPase. Sodium potassium ATPase is always going to be dragging sodium outside of the cell and moving the potassium to the inside of the cell. And this, in order to take place, it requires ATP. It requires you to hydrolyze ATP. This is a form of energy that we're going to be consuming. Questions? Questions? All right, so looking here at this action potential that we've just created, drawing it again here. Graph, on this graph, we are plotting time and voltage. So we started by the resting membrane potential on here, negative 70 millivolts. I've got a stimulus at this point, and this stimulus allowed enough sodium channels to open. This creates the depolarization phase of the action potential. Once I reach the apex of this wave, what's going to happen? I close sodium channels and I open potassium channels. Allowing the potassium out will make the membrane to return back to its resting membrane potential. We call this phase as my repolarization phase. Remember, if you kept moving more potassium outside of the cell beyond the resting membrane potential, this will create a state of exaggerated polarity. We call this phase as my hyperpolarization phase. And remember, why do I have hyperpolarization? Because simply I will be moving more potassium out through the potassium channels, which are slow to close. So when would I be able to respond to another stimulus? Here, 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 here. When? If I give you numbers on here, 1, 2, 3, 4, and 5. So when would I be able to respond to another stimulus? If I get the stimulus at this point, when I am depolarizing like this, or when I get it during later depolarization, or if I get the stimulus during early repolarization, or if I'm getting the stimulus during late repolarization, or if I'm getting the stimulus during hyperpolarization. What is the earliest time that you can respond to a stimulus? 5, or you're gonna wait until you get back to the resting membrane polarity after you return back to the resting membrane polarity after you're done with the hyperpolarization. Number 6. When would I be able to respond to a stimulus? 1, 2, 3, 4, or 5. All right, so please write your answer as your first in-class activity for today. All right, 4, 4, 5, 4, 6. So what is the state of the channels? Can you remind me? During depolarization, which channels are open? During depolarization, here again, which channels are going to be open? Sodium channels, right? And how do you respond to a stimulus? How would I respond to a stimulus? I open sodium channels, right? So if I'm telling you here, during depolarization, sodium channels are open. Can I open a sodium channel which is already opened? Can you do anything to this channel if it's already open? Can you open it if it's already opened? No. So what will be your response here to this stimulus? You're gonna neglect it. Same during late depolarization, nothing gonna happen. How about early repolarization? When I start to repolarize, what is the state of the channels? What's the state of the channels? During early repolarization here at this point on the graph. I open potassium channels. I open potassium channels and the potassium is traveling to the outside, right? Do I do anything for the sodium? Yes, you close the sodium, right? So during early repolarization, what am I doing? I am closing the sodium and I am opening potassium. So what's going to happen to this stimulus that you've just received during repolarization? Do you think would you be able to respond? Would you be able to respond? Can you open? How do you respond again? Remind me, how do you respond to a stimulus? You open sodium channels. So can I open and close the same door in the same exact moment? Can you open and close it in the same time? Can you tell a person, can you please open and close the door in the same exact second? No. So would I be responding to this stimulus that I've received while I'm closing the sodium? Can I open it while I'm closing it? No. So it would be neglected. As if you didn't receive the stimulus. How about during late repolarization and hyperpolarization? During late repolarization and during hyperpolarization, what is the state of the sodium channels? They are already closed. They are closed. I'm not closing them right now. They are already closed. And what is the state of the potassium channels? They are opened. Can I respond to a stimulus that I will be getting during late repolarization or during hyperpolarization? What do you think? What do you think going to happen to those 4 and 5? So definitely 6, you're going to respond. During 4 and 5, what is going on in here? You have sodium channels which are closed, and you have potassium channels which are opened. Would you respond to the stimulus? Would you create another electrical impulse here? Like this. What do you think? Yes or no? Would I respond? Guess, guess, guess. Yes, yes, yes. Only 3 can guess here. No. So I didn't say it won't. I didn't say it won't. I'm asking you, will it or not? As a guess. So for number 5, I didn't say it won't create an electrical impulse. It won't respond. I'm not saying that it won't respond. I'm asking you, do you think it's going to be responding to the stimulus if received in 4 and 5 or not. And I did explain to you that during 4 and 5, what's going on in here in the sodium and potassium channels, sodium channels are closed and the potassium channels are still open. All right, so 4 guesses. What do you think? Yes. It's neither yes or no. It's neither yes or no. It's according to what? According to the strength of the stimulus. If I received just a threshold stimulus, let's say, in order for me to depolarize, I did need to allow 50 sodium inside the cell. All right, for me to depolarize, just a number. If I'm telling you, in order for you to depolarize, you need to get 50 positively charged ions to move from outside of the cell to the inside of the cell. All right, if you're starting from negative 70 millivolts. What if I'm telling you that now you are moving some positively charged ions outside of the cell? You are during repolarization. Potassium channels are moving outside of the cell. So if you give me a threshold stimulus that allows only 50 sodium to travel in, would this be enough for me to depolarize? I will tell you the example in a different way here. So let's say you are satisfied having 1,000 bucks in savings, let's say per month. All right, you are satisfied having 1,000 bucks in your saving accounts to be added every month. So this is the action potential, your satisfaction. This is the action potential. You are satisfied. What was the threshold stimulus, the minimal stimulation that will make you satisfied, the minimum amount of money that you need to keep in your savings account each month for you to become satisfied, for you to have an action potential? It's 1,000 bucks. But now you've got a baby and you're gonna be spending 500 bucks from this amount that you were supposed to get in your savings account. Each month you're gonna be spending those 500 bucks just for the expenses, extra expenses because you've recently got a baby. Would you be satisfied? Would you create an action potential? Your satisfaction here is the action potential. Would you be satisfied if you are spending 500 and the amount that you should be depositing was 1,000, so now you're only depositing 500, the remaining 500, because your expenses have been going up and you are spending out of those 1,000 bucks 500. Would you be satisfied if I told you that your minimal satisfaction is gonna be met when you put 1,000 bucks in your savings account? So you won't be satisfied. So there will be no action potential. So what you should be doing in order for you to become satisfied, putting into consideration that there is no returns for a baby. So you can't return the baby back. All right. No exchange or return for the baby. So you should be gaining an extra 500. So rather than having an extra 1,000 each month, you should be getting an extra 1,500 for you to get the 1,000 bucks in your savings account and be able to spend those 500, which are your expenses. All right, at this point only, you're gonna be satisfied. You're gonna be able to generate an action potential. So let's apply this same concept on the sodium. So let's say, in order for me to create an action potential, I need to get 50 sodium in. Or let's forget about the sodium. 50 positively charged in. 50 positive charges in. During late repolarization, I am losing some of the positive charges. So let's say you're losing 25 positive charges. You are now experiencing more expenses. So I'm losing positive charges. So what you should be doing, would you be able to create an action potential? Would you be satisfied having only 50 positive charges in? Would this create an action potential for you? If you are currently losing 25 to the outside of the cell, what do you think? Would you be able to create this depolarization? No. So what you're gonna need to do, you need, rather than moving 50 positively charged in, you need instead, because you are losing 25 positive charges out, you're gonna need to allow 75 positive charges in. Exactly. So this means what for me? This means I need to get a stronger stimulus in order for me to respond. What if you've got a threshold stimulus, the ones that will give you only 50 positive charges in? Would you be responding? Would you create an action potential? Would you depolarize? No. What if you got a strong stimulus greater than the threshold stimulation? Yes, I will respond. Make sense? Make sense? So what do we call the time during which you want to respond to any kind of stimulation, either threshold or greater than threshold? And this is going to be during depolarization and early repolarization. Again, why you want to respond to any kind of stimulation? Because how do you respond? You open sodium channels. You open sodium channels. And if the sodium channels are already opened during depolarization, or you are currently closing them, you will not respond. We call this phase in which I'm not going to be responding to any kind of stimulation. It's my absolute refractory period. Absolute. You won't respond to any kind of stimulation, regardless of how strong this stimulus is. Absolute refractory period. How about during late repolarization and during hyperpolarization? Sodium channels are already closed and I am leaking some of the potassium out. So I need a stronger stimulus. I will not be responding to a threshold stimulus, but I will be able to respond to a stronger stimulus. So what do we call this phase? We call it a relative refractory period. It's relative. It's not something absolute. It's relative. It's according to what you're going to respond or not. It's according to the strength of the stimulus. Again, what is the refractory period? This is the time in which you won't be responding. We have two types of refractory periods. We've got the absolute refractory period. Absolute refractory period. This is the time during which you won't be able to respond to any kind of stimulation. The cell here in this condition cannot be stimulated. Why I can't be stimulated? Because simply my sodium channels either are already open or I'm currently closing them. So I can't open an already open channel, and I can't open and close the same channel in the same exact time. So I won't respond. I will neglect this stimulus. Compared to the relative refractory period. In the relative refractory period, the membrane can be stimulated by higher intensity stimulus. Why higher intensity stimulus? Remember, your sodium channels are already closed, so you can't open them, you can be stimulated. The door is closed. Can you open a closed door? Yes, I can open a closed door. I have no problem with it. But what is going on with the potassium channels? Potassium channels are also open. They are leaking potassium outside of the cell. And if I am leaking potassium channels outside of the cells, this means I'm losing positive charges. So I need to not open only the minimum number of sodium channels, I need to open a greater number of sodium channels. And what allows me to open a greater number of sodium channels is going to be a stronger stimulus. A higher intensity stimulation than the threshold. Easy, easy. This is highly, highly important for you to be able to understand the cardiac physiology later on with AP2 if you're planning to take AP2. Right, questions? All right, so we can have a break for 10. We come back.
I didn't get that. Could you try again?
So we can have a break. I'm not sure what's going on here. All right, so we can have a break for 10. We get back and we will review the PowerPoint presentation covering the action potential and the absolute refractory period. And we're going to be starting our next chapter, which covers the CNS as well. All right, so anybody has question before we go for a break. Questions? All right, so we'll see you in 10. All right, so welcome back. For those who are asking about the answer of the question, the answer is going to be an essay. So you're going to be mentioning that for 1, 2, and 3, the cell can be stimulated. It's absolute refractory period. And for 4 and 5, cell only responds to a high intensity stimulus, and for 6, cell gets stimulated by a threshold stimulus. As usual. All right. This answers your question, Era. This is our first question for today. All right, so you're gonna tell me that for 1, 2, and 3, cell can be stimulated because it's an absolute refractory period. For 4, 5, cell responds to high intensity stimulus. It's a relative refractory period. And for 6, the cell will be stimulated normally. So a threshold stimulus can stimulate the cell. All right, so let's go over here the membrane potentials. The neurons are highly irritable cells. They will respond to adequate stimulus, and again, the adequate stimulation here is going to be our threshold stimulus. And in response to the threshold stimulus, you will be able to create a nerve impulse. Impulse is always the same regardless of the strength of the stimulus. It's an all or none. Whether you create a stimulus, if you've got enough, you create an impulse if you've got enough stimulation, or you don't create an impulse. It's an all or none signal, whether you have it or not. The resting membrane potential is going to be the potential difference between the inside of the cell membrane and the outside of the cell membrane, and again, it's equal to negative 70 millivolts. Cytoplasmic side is going to be more negatively charged compared to the outside. This is going to be generated by the difference in ions inside and outside of the cell and the differential permeability of the plasma membrane. So plasma membrane is going to be more permeable to ions compared to others. Higher concentrations of sodium and chlorine outside of the cell. I have higher potassium concentrations inside of the cell. And a lot of the anions are going to be located inside the cell, which will make the inside of the cell more negatively charged. Sodium potassium ATPase pump will allow you to maintain the balance, keeping the sodium at higher concentrations outside of the cell and potassium at higher concentrations inside the cell. Potassium is going to be easier to pass through the cell membrane. The cell membrane is going to be 75 times better in terms of potassium conduction rather than compared to sodium. So potassium is 75 times easier to travel through the membrane compared to the sodium, which will allow the potassium to accumulate within the cell. Positive charge develops outside the cell, and negative charge develops inside of the cell. So inside during rest is more negatively charged compared to the outside, which is going to be more positive charged by the effect of the ion distribution. Here's this diagram is showing you that you have much more potassium inside the cell and much more sodium outside of the cell. Membrane potentials will change the concentrations of ions across the membrane. Permeability of the membrane to ions also changes by opening or closing channels. Changes in the membrane potentials are signals used to receive, integrate, or send information. Those are the electrical signals that you're going to be conducting through the nerve cells. We've got two types of signals. We've got short distance signals. Those are localized currents that won't be propagating. So I will be depolarizing, let's say, this point, but I didn't open enough sodium channels for me to send out this signal throughout the length of the nerve cell. Compared to action potentials, those are long distance signals that will be traveling along the length of the axons. If you remember, the axons are those short, those long processes of the nerve cells. Depolarization is a reduction in the membrane potential. So you're starting from -70, and if you are depolarizing, this means you're going towards the zero. If you're starting from a -70 millivolts, so when you depolarize, the inside will become more positively charged by allowing the sodium in. So inside of the cell will become more positively charged. This means that it will be moving towards the zero. This is what we call depolarization. Compared to a hyperpolarization, you are starting again from -70 millivolts. This is your resting membrane potential. But rather than opening sodium, you did open potassium. So what's going to happen? Potassium is going to be moving outside of the cell, and the membrane will become more negatively charged on the inside. It's going to be even more negative than your resting membrane potential. So you see on here, I started by -70 millivolts, and as I open the potassium channels, this allows the efflux of potassium, making the membrane more negatively charged than resting membrane potential. Graded potentials are going to be localized changes in the membrane, either in the form of depolarizations or hyperpolarizations that won't be propagating for long distances. So I've got a stimulus. It's not a threshold stimulus. It's lower than threshold. So I will open some sodium channels, but not enough sodium channels for the voltage-gated sodium channels to open and the electrical impulse to propagate. So it's going to be a localized current only at this point in the cell. An action potential, on the other hand, it's going to be an all-around. It does not decrease in magnitude over distance. So the action potential at the beginning of the axon is going to be the same as at the end of the axon. So there will be no change because every time you're going to open enough sodium channels, creating the same change in the membrane polarity, making the inside again more positive compared to the outside, which will become more negatively charged. It has the same magnitude throughout the length of the nerve fiber. This is going to be the type of signals that you're going to be conducting for long distances along your nerve cells. Phases here of the action potential, we started first by resting membrane potential, which is around -70. You open sodium channels, you allow the sodium in, the inside of the cell will become more positively charged. This is my depolarization phase of the action potential. When I reach 30 millivolts, this will be allowing the sodium channels to close, if you remember, and will allow the potassium channels to open, allowing potassium out. And if I'm losing potassium outside of the cell, potassium efflux, this means the inside of the cell membrane will become more negatively charged. So you're going to be returning back to your resting membrane potential. You see on here, if you kept going, and you will, in most of the cases, you will be leaking more potassium even beyond the resting membrane potential. So even after I reach -70 millivolts, I will still be leaking potassium outside, which will make the inside of the membrane to be more negatively charged than the resting membrane potential. All right, we call this phase as my hyperpolarization phase. All right, this is a diagram showing the steps that we've discussed. Again, in a resting state, if I don't have any stimulation, sodium channels, the outer gate of the sodium channel is closed, potassium channels are closed. When I receive enough stimulus, enough stimulation to the excitable cell, to the nerve cell on here, what's going to happen? I will open sodium channels, allowing the sodium in the cell. So the inside of the cell will become more positively charged. This is what we call the depolarization. After I reach 30 millivolts, what's going to happen? Sodium channels will close, preventing any further influx of sodium. And you're going to be opening the potassium channels, allowing the efflux of potassium. So the potassium now is able to leave the cell. After I return back to my resting membrane potential, -70 millivolts, the potassium channels will not close. They will still be leaking some potassium outside of the cell, making the inside even more negatively charged than the resting membrane potential. This is what we call the hyperpolarization. All right, for the second question of the in-class activity for today, it's going to be the resting state of a neuron is called A, B, C, D, E, depolarization, resting membrane potential, repolarization, hyperpolarization, or action potential. Which one do you think? resting membrane potential, exactly. Remember, the action potential is an all or none. Whether I have full strength or it's not gonna be created. So if you receive the stimulus lower than threshold, would you form an action potential? No. How about getting a threshold stimulus? Would I get an action potential? Yes. What if I get stronger than threshold? Would I get action potential? Yes. What's the difference between the action potential that you get with a threshold to stimulus and the action potentials that you're going to be creating to a higher intensity stimulus? Is there any difference between the two action potentials that you're creating? No difference, no difference. But how would you know that this sound is louder, that this red is brighter? How would I know that there is a stronger stimulus if the same signal gonna be sent in both conditions? How would I be able to figure out that, yeah, this is stimulus, yeah, this is moderate pain, and this is very strong pain? How would I be able to know the difference between the two if both of them gonna be creating the same exact action potential? Frequency of, not stimulation of, action potentials that you're gonna be creating. So getting exposed to much stronger stimulation results in not an increase in the amplitude of the action potential, but in the formation of more action potentials. More action potentials that will be formed. So your brain will be decoding, yeah, I've got, let's say, from a pain receptor, I've got 10 stimuli. And in another situation, I've got 2. So 10 stimuli means I've got a stronger 10 action potentials that I'm receiving. This means that I've got a stronger stimulus. Moderate pains that I'm detecting. I'm detecting this because I did receive only, let's say, 2 action potentials. So coming from the pain receptor, I did detect 2 action potentials. So this is a moderate amount of pain. How about if I did have a stronger stimulation of the pain receptors? The brain is going to be receiving 10 action potentials, let's say. So you're going to be translating, getting 10 action potentials from the pain receptors. This is a very strong pain. So those are not accurate numbers. Those are just for you to get the idea. Questions? All right, so again, what is the refractory period? This is the time when the membrane is not going to be responding to a threshold stimulus. It might be an absolute refractory period during which you won't be responding to any kind of stimulation, either threshold or higher intensity stimulus. I won't be responding. Why? Because this is going to be taking place during depolarization or early repolarization. And during depolarization, the sodium channels are open. And during early repolarization, you're closing the sodium channels. You can't open an open door, and you can't open and close the same door at the same time. So you won't be responding to any kind of stimulus. This is an absolute refractory period. How about in a relative refractory period? In a relative refractory period, this is going to be during late repolarization and hyperpolarization, which you already have closed the sodium channels, so you can't open them. You can't open them now. They are already closed. But in the same time, you need to put into consideration that you're leaking potassium outside of the cell. So if I'm allowing potassium outside of the cell, this is a positive charge that needs to be substituted. So if I will be creating an action potential opening 50 sodium channels, in order for me to replace the amount of potassium that I am losing outside of the cell, I need to open an extra 25 sodium channels to get more sodium for me to form an action potential. All right, so it's going to be relative. Your response is relative. Relative to what again? To the strength of the stimulus. If you got threshold, you won't respond. If you got a higher intensity stimulus, you're going to be able to respond to the stimulus by creating an electrical impulse. What affects the conduction velocity? How fast am I conducting the electrical signal? First is going to be the diameter of the nerve fiber. The larger the diameter of the fiber, the faster the electrical conduction will be. Another thing is going to be the state of myelination, the effect of myelination. If you remember, when we studied the structure of the neuron, we've mentioned that the axons were surrounded by myelin sheaths. And if you remember, not all nerve fibers are going to be myelinated. We mentioned last class that we've got unmyelinated nerve fibers. Remember, what was the function here of the myelin sheath? First, as we stated last time, it was the insulation. The second function was to something that we didn't mention, I kept it for today's topic, is increase the conduction velocity. How do I increase the conduction velocity? Again, what is the action potential? Can you remind me? You change the membrane polarity. Inside was more positively charged, outside was more positively charged, inside was more negatively charged. You flip the charges, inside becomes more positive, outside becomes more negative, and then you return it back. This is your electrical impulse. This is briefly the electrical impulse. Depolarize, then repolarize. So this takes place where, if you remember, across the plasma membrane of the cell. If I told you that this is an insulator, will it allow electricity? The myelin sheath. If I'm telling you that the myelin sheath is an insulator, will it allow electricity to be conducted? No. It won't allow electricity to be conducted. So how would I conduct the electrical impulse along the plasma membrane? Remember, the myelin sheath is not gonna be fully covering the nerve fibers, the axons. We did have those interruptions in the myelin sheath. If you remember, we call those are the nodes of Ranvier. So you can create an electrical impulse, an action potential only in the gaps between the Schwann cells, because the myelin sheath will not allow you to create an electrical impulse at this point, on those points covered with Schwann cells or surrounded by the Schwann cells. All right, so what I will do here, if you check a nerve fiber like this, you compare, this is an unmyelinated nerve fiber. This is the myelinated nerve fiber. It has myelin sheath on top of it, like this. So you are changing the membrane polarity. This is the action potential. So you're going to change the membrane polarity at this point. So it propagates to the next point, propagates to the next point, propagates to the next point, next point, next point, next point, next point. Ton, 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 ton. I've arrived. How about if I'm conducting the electrical signal along a myelinated nerve fiber? I can only create the signal at the sites where I don't have myelin sheath covering. So I create an electrical signal and then boom, you can't allow the electrical signal to pass through this part of the membrane. So you're going to be jumping exactly over the myelin sheath. You're going to jump from one node of Ranvier to the next node of Ranvier. Boom, boom, boom. Which one was faster, one or two? So again, in number one, boom, 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 boom. For number two, boom, 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 boom. Which one is faster? Two. So what is the effect of the myelination here? What is the effect of myelination? Speeds up the velocity of the electrical conduction along the nerve fiber. Easy, easy. All right, so you see on here, this is difference in terms of electrical conduction along an unmyelinated and a myelinated nerve fiber. Right, so here you need to create the action potential. You need to open the channels, close the channels, open the channel at every single segment of the length of the fiber. But compared to here in myelinated nerve fibers, you're going to be only opening and closing the channels at the nodes of Ranvier, because simply those parts are covered with myelin sheaths, which won't allow the electrical conduction. We're going to be jumping from one node of Ranvier to the next node of Ranvier, allowing faster electrical signal conduction. All right, what's a synapse? A synapse is going to be the connection between a neuron and another neuron, or a neuron and the effector organ. So like what we've seen was the neuromuscular junction, if you remember, we did have this terminal end of the nerve fiber. It's going to be connected to the muscle cell. And remember the connection here, the gap in between the two, this is my synapse. So if I'm connecting two nerve cells to one another, the one that will be located before the synapse, this is my presynaptic neuron. The one that will come afterwards is going to be my postsynaptic neuron. So this is going to be my postsynaptic neuron. So how can I connect two nerve cells to one another? I can connect the terminal ends of the neuron to the cell body of another neuron. So we call this is an axosomatic. Soma means body. So if you're connecting the axon of a nerve cell to the cell body of another nerve cell, it's an axosomatic synapse. If I'm connecting an axon of a nerve cell to the dendrites of another nerve cell, we call this as an axodendritic synapse. Axodendritic. What if you're connecting an axon to the axon hillock of another nerve cell? We call this as a connection between two axons, so we're going to call it an axonic synapse. Again, what do we call the cell, nerve cell that will be located before the synapse? It's going to be my presynaptic neuron. Well, the one that comes after the synapse, it's my postsynaptic neuron. Any questions? Any questions? Anybody is still awake? Yes. No questions? But nobody is still awake. All right, great. How many of you are coming from work? So you're attending after you're done with work? 2, 3, 4. All right, all right. And who is not coming from work? We'll see here. All right. So moving on to the chemical synapse. Chemical synapses are going to be the connections between the two nerve cells in which one nerve cell, the presynaptic neuron, is going to be releasing the chemicals, and those chemicals are called my neurotransmitters. They will have an effect on the postsynaptic neuron. So if I'm drawing two neurons that are connected by a synapse like this, this is my presynaptic neuron, this is my postsynaptic neuron, and this gap in between is my synapse. So in chemical synapses, we've got neurotransmitters that will be released from the axon terminal of the presynaptic neuron and will be binding to the receptors, to their specific receptors, this time on the postsynaptic neuron. The synaptic cleft is this fluid-filled space between the presynaptic and the postsynaptic neuron, and this prevents the nerve impulses from directly passing from one neuron to the next. So if we're looking here, this is my nerve cell like this. It's connected to another nerve cell. Those are my pterodendria. Those are my axon terminals. And I've got another nerve cell waiting to get the signal. This space again is what we call the synaptic cleft. So the chemicals will be released from the axon terminals of the presynaptic neuron. And they will bind to the receptors located on the receptive region of the postsynaptic neuron. Why do I have this space filled by fluid between the two nerve cells? Why would I be releasing a neurotransmitter and this neurotransmitter binds to a receptor on the postsynaptic neuron, and this allows the opening of the sodium channels for you to create an electrical signal that will be traveling along the postsynaptic neuron. You remember here when we discussed the neuromuscular junction, we've got an electrical signal traveling down, and this electrical signal will stimulate the calcium channels, voltage gated calcium channels to open, and the influx of the calcium is what triggers the neurotransmitter vesicles to fuse with the cell membrane, releasing the neurotransmitters. And if you remember, the neurotransmitter, the example that we've seen for the neurotransmitter was the acetylcholine and the neuromuscular junction. Here, we've got the neurotransmitters that will also bind to its receptor, and when it binds to the receptor, it allows ions to travel in and out, causing the creation of an electrical impulse that will be traveling for a long distance, or it's going to be remaining according to the number of those channels that you did open. And we did justify the presence of the synaptic cleft by saying here that this synaptic cleft is going to be beneficial to prevent any direct passage of an electrical impulse from one neuron to the next neuron. Why do you want to do all this? Why do you want to release chemicals, those chemicals to bind to the receptors, to open the channels, to create another signal that travels along the postsynaptic neuron? Why you don't simply allow the electrical impulse to travel directly from the presynaptic neuron to the postsynaptic neuron, and that's it? Why would I do this? Why would I create why would I release neurotransmitter that will bind to the receptor to open other channels to create an electrical impulse? If I did already have an electrical impulse, why I just simply don't allow it to travel directly from one cell to the next cell? Any guesses? Protective? If I'm saying, so for faster, it won't be faster, theoretically speaking, if I'm conducting just the electrical signal without having to release chemicals and bind and open. So if I'm just conducting the electrical signal, it's going to actually be faster. Why would I need an insulation here? I don't want to have an insulation. I want the signal to be conducted from one cell to another. Variety of sensations. Variety of sensation and cooling. So are all the neurotransmitters going to be acting the same way? What do you think? No. So in some cases, you want to send an excitatory impulse to the postsynaptic neuron. So I want to create an electrical signal for it to become activated and to affect the effector organ. But what if I am trying to send an inhibitory impulse, or I'm trying to inhibit the activity? What is the kind of signals that I will be delivering? It's still an action potential, but I will be releasing a different type of chemical to affect the same exact cell. But this time I will not be creating an electrical impulse, but I will create a hyperpolarization state. So I will make it harder for the cell to send an excitatory impulse, lowering down the sensitivity. Exactly. So like in the heart, for example. Can you remind me what part of the nervous system is going to be responsible to regulate the heart rate, the force of contraction of the heart? Autonomic, exactly. One part of the autonomic nervous system is called the sympathetic, and the other part is called do you remember? Parasympathetic, exactly. So one is supposed to increase the heart rate. One is supposed to reduce the heart rate. through the two nerves that I'm getting here, I'm conducting the same action potential. Either you want to increase the heart rate or you want to reduce the heart rate. It's the same electrical impulse. The difference is in here, is what kind of neurotransmitter are you gonna be releasing? Am I gonna be releasing norepinephrine, or am I gonna be releasing acetylcholine? What is the difference? This one gonna open sodium channels. This means what? It's gonna be creating a depolarization. So let's say if I'm starting from -50 on here, or let's say, let's keep it, I don't want to get you confused with the numbers. So let's start as if we're dealing with a nerve cell. So we're starting from -70. If I open sodium channels, will I be moving up or down? What do you think? Up one, down two. If I open sodium channels, I am moving up, right? I'm becoming more positively charged. So if I did release norepinephrine, I will open sodium channels. The inside of the cell membrane is going to become more positively charged. So will it be easier or harder for the heart to create a signal? So rather than starting from -70, let's say I'm starting from -30. Rather than starting here at this point, I will start here at this point. So I will take less time to create the electrical signal. And as I take less time to create the electrical signal, what's going to happen? I can create more electrical impulses. And for each of those electrical impulses, remember, your muscle responds by contracting. So I can create faster contractions. How about if I open, under the effect of acetylcholine, I opened potassium channels. So the potassium is going to be leaving the cells. So where would I be where would I be starting? From -70, from below -70, a more negative point, like -90, let's say, or from -30, something less negative. So one here, -90 millivolts, or -70 or -30. Which one? If I did open potassium, you are making the inside of the plasma membrane to become more negative. So you're going down. I'm going down below -70 millivolts. So if I'm starting from this point, from -9, will it take me more or less time to depolarize compared to if I'm normally starting from -70? Would you take more or less time? More time for each depolarization. If you're taking more time for each depolarization, this means you have the ability to create less depolarizations per minute. And as a result, you're going to have, in response to each of the depolarizations, you're going to have a contraction. So if I have less depolarizations per minute, I have less cardiac contractions per minute. This lowers down my heart rate. Make sense? We're going to discuss this in in much more details when you study the topic in AP2. But just to give you an impression about why would I need a neurotransmitter in between the cells, because not every neurotransmitter gonna have the same action. So in some cases, I might be triggering a depolarization, I might be triggering a hyperpolarization state. So the neurotransmitter is not going to essentially be inducing the formation of another similar electrical impulse. All right. Any questions? Any questions? All right, so let's stop at this point where we can be starting next class. All right, make sure you make it to our next class. We're gonna be covering a lot of the CNS, the structure of the brain, which is essential for the upcoming lab exam. So please also make sure that you complete your muscles lab exam. It was available starting from last Tuesday. It will be available for 1 week. So please make sure to not wait until the due date to get it completed, because I will not be reopening the quiz if you missed the due date. You've got more than enough time to get it completed. All right, please also make sure you are using a desktop or a laptop, not from a tablet or from a Chromebook. Also, try to use Google Chrome as your internet browser. Some people did report that the pictures, the diagrams on the test are not loading, and it might be because of some issue with the browser itself. All right, so please avoid using devices that don't have a complete desktop operating system. Also, please make sure you are using Google Chrome browser while you are taking the exam. I'll tell you much more information about the last lab exam on our last meeting, on our upcoming meeting on Tuesday next week. Until then, please, MacBook should work, yes. MacBook, I personally don't have any PC anymore. And I was able to access I was able to access the exam in a student view. So I'm not sure what was the issue. Some of the people who did report that they didn't have their images or the diagrams loading, some of them didn't get in contact back with me and tell me what happened with them. So I'm not sure. So they sent me an email with no pictures, and they told me we couldn't find the pictures, or we didn't find the diagrams on the test. When I replied, they never get back in contact with me for me to figure out what was the problem. All right, so just try to use a Google Chrome operating system. Also, if you have any ad blockers, if you have pop-up blocker, turn them off while you are taking the test, because it might be a pop-up blocker that prevents you from loading the diagrams. All right. Any questions for me? Any questions? All right, great. Thank you so much for making it to the meeting today, and we'll see you next week. I hope you have a good weekend. Stay safe. Thank you. Thank you so much.